All right, so we're very happy to have Dr. Zachary Freyberg. He's an MD, PhD, who is currently at the University of Pittsburgh in the departments of psychiatry and cell biology. His talk today is development of novel tools for dissection of central versus peripheral dopamine D2-like receptor signaling and dysglycemia. Just a little bit of a background for him is that since arriving at the University of Pittsburgh in 2016, he has been an independent investigator using comparative approaches in Drosophila mouse and postmortem human models to study the mechanisms by which synaptic vesicles load, retain, and release dopamine in the central nervous system and in the periphery. This work examines both mechanisms of psychostimulant action as well as presynaptic neurotransmitter co-transmission and its emerging relevance to dopamine neuron vulnerability and degeneration. In parallel, Dr. Freyberg is examining the roles of dopamine and dopamine receptors outside of the CNS in the periphery and their roles in glycemic control. The talk presented here is going to describe the development of a new family of peripherally limited drugs that selectively target dopaminergic D2-like receptors outside the brain. And the rationale of these studies was to dissect the functional relevance of dopamine signaling in the brain versus metabolically relevant peripheral organs, including the pancreas and connecting dysglycemia. The ultimate goal of this work is to better understand the mechanisms by which peripheral dopamine functions as a metabolic receptor. And this can be applied towards the development of novel mechanisms to treat diabetes, as well as to treat the metabolic disturbances produced by antipsychotic drugs. And I'm just going to drop a couple of his recent papers in the chat for those who have interest. And thanks so much for joining us. This is really going to be an interesting talk.
Oh, it is such a pleasure and honor. I'm really, really grateful to be invited today, for being able to present some of this work and just being here. And the work that Sugar Science does is such an amazing thing, and it's really helping the community. Like I said, I'm just humbled to be here. So
Thank you very much.
Sure. So as Dr. Wesley described, the title of the talk today is Development of Novel Tools for Dissection of Central versus Peripheral Dopamine D2-like Receptor Signaling and Dysglycemia. So fundamentally, right, this is going to be a talk about dopamine and dopamine receptors and their relevance to metabolism and metabolic control. So before I begin and really get into the heart of the talk, I just want to orient people in terms of a lot of the terminology that I'm going to be using. So first of all, what is dopamine? Dopamine is a so-called neurotransmitter. It's a derivative, actually, of the amino acid tyrosine. It's modified intracellularly to form dopamine, which in turn then signals through receptors, G protein coupled receptors that are expressed classically on the cell surface. And these dopamine receptors are traditionally separated into two general families, D1-like receptors versus D2-like receptors. The D1-like receptors are known as stimulatory dopamine receptors. Why? Because D1 and D5 receptors, which constitute this family, are coupled to stimulatory G proteins. These are intracellular effectors. These G proteins, such as G alpha S, S for stimulatory, stimulate the production of cyclic AMP, or CAMP. And this cyclic AMP, as it rises in the cell, facilitates or promotes the release of hormones, such as insulin. On the other hand, you have a second family of dopamine receptors known as D2-like receptors, and the bulk of the talk is going to focus on this family. And these D2-like receptors are known as inhibitory dopamine receptors. Why? Because they're coupled to G alpha I, or inhibitory G proteins. And these G proteins inhibit production of cyclic AMP, and then by definition, modulate the decrease in resulting hormone secretion. So stimulation of D2-like receptors diminishes activity-dependent hormone release. So these dopamine receptors are best studied in the brain. The vast majority of work studying dopamine and dopamine signaling is in the context of the central nervous system, or CNS. And interestingly, clinically efficacious antipsychotic drug treatment of disorders, mental disorders such as schizophrenia or bipolar disorder or major depression, target this dopamine system. These psychiatric medications are blockers of these dopamine D2-like receptors. However, both dopamine and dopamine receptors are also expressed outside of the central nervous system. This is something that really, at least in neuropsychiatry, hasn't been appreciated. So the dopamine itself, now the ligand for these receptors, is thought to be primarily a neurotransmitter in the central nervous system, in the brain. And you can kind of guess that from the first syllable of the word neurotransmitter. But ironically, the largest pools, repositories of dopamine, are actually outside of the brain and the periphery. The gut and the kidney are the largest pools of dopamine in the body, followed by the pancreas. It seems that the brain is actually the late guest to the party. The brain actually has considerably smaller pools of dopamine and dopamine production compared to these other systems in the body. In the gut, dopamine promotes digestion and metabolism. In the kidney, dopamine is absolutely essential for blood pressure control. And in the pancreas, which is something I'll be talking about shortly, dopamine is absolutely critical for glycemic control. Moreover, in terms of trying to tease out dopamine's precise roles in metabolic regulation, the literature actually has provided us with a lot of important hints. So for example, L-DOPA, which is the precursor for dopamine biosynthesis, and one of the cornerstone treatments for Parkinson's disease, is a metabolic modulator. In fact, if you go back in the early Parkinson's literature, when Parkinson's patients were initially prescribed L-DOPA for the treatment of their motor symptoms, they became hyperglycemic. Their blood sugar levels rose, suggesting that L-DOPA and its conversion to dopamine was somehow connected with glycemic control. More recently, dopamine D2-like receptor agonists, drugs which stimulate dopamine D2 and D3 receptors, including bromocriptine, themselves actually affect glycemic control. In fact, D2, D3 receptor agonist bromocriptine was actually FDA approved to treat type 2 diabetes. But why? Why is it that targeting these dopamine D2-like receptors has effects on glycemic control and therefore on systemic metabolism? I just told you a minute ago that these receptors are not, and dopamine for what it's worth, are not only produced and expressed in the brain, but also in the periphery. So is it possible that the metabolic effects of dopamine signaling are primarily driven by the effects in the periphery? Or is it that they are primarily driven by their metabolic effects in the brain? Because indeed, they're expressed in regions of the brain such as the hypothalamus, which themselves regulate systemic metabolism. Or, frustratingly, is it some combination of the two with ongoing interplay? And so this is really our starting point for this story. Now, to give you just a little bit more background, in terms of dopamine D2 receptor targets in the periphery, for my journey in this story, it kicked off with the publication of a paper by Pierre Michelet's group at the University of Geneva in 2005, demonstrating that dopamine D2-like receptors are expressed in pancreatic beta cells. And in fact, these D2 receptors, these targets of antipsychotic drugs, of many other psychiatric medications, are actually localized to the same secretory granules that load insulin, suggesting that there is a relationship between the two. And indeed, unlike neurons, in fact, these pancreatic beta cells express all 5 dopamine receptors, D1, D2, D3, D4, and D5, as well as the biosynthetic machinery responsible for dopamine production. which leads us to the next question. What are the actual sources of pancreatic dopamine? So the first thought is, well, dopamine is classically a neurotransmitter. So the nervous system must be innervating a peripheral organ like the pancreas and providing dopamine through neuronal biosynthesis. Well, dopamine is a catecholamine. It's the precursor for norepinephrine, which we know is released by sympathetic neurons. And sympathetic innervation of mouse and human endocrine pancreas definitely is known to have an effect in terms of regulating glucose-stimulated insulin secretion. Yet, more recent literature has demonstrated that sympathetic innervation of both the mouse and human pancreas is actually much sparser than previously thought. And as I mentioned a minute ago, whatever neurotransmitters the sympathetic neurons are releasing locally is actually noradrenergic. It's norepinephrine, not dopamine. And indeed, there's no known extrinsic source of dopaminergic innervation of islets. So what gives? So is it possible then that if local innervation of the pancreas is not providing dopamine for these dopamine receptors, that in fact, the dopamine is actually coming from within rather than from without? That these islets are making their own dopamine. And indeed, that's what our experimental evidence suggests. We took islets and immortalized beta cells, and we just gave them L-DOPA. And what we found was a dose-dependent relationship of L-DOPA-mediated inhibition of glucose-stimulated insulin secretion. So the more L-DOPA you feed beta cells in islets, the greater the inhibition of glucose-stimulated insulin secretion. Now, is it that the L-DOPA itself is driving these effects, or is it its conversion to dopamine and therefore signaling through dopamine D2-like receptors? So what we did was we actually blocked the conversion of L-DOPA, which you can see on the red curve here, with a blocker of the enzyme that converts L-DOPA to dopamine called AADC, aromatic amino acid decarboxylase. So when we block this conversion, we got no further inhibition, which you see in the black curve, suggesting that these islets take up dopamine precursors like L-DOPA, which circulate in the bloodstream, which actually rise more than 50-fold after you eat, and convert the L-DOPA to dopamine locally within islets. And post-conversion, that locally produced dopamine then signals through dopamine receptors to modulate insulin secretion. And indeed, moving beyond precursors, when we just do dose response curves in both human and mouse islets with dopamine itself, we can recapitulate our L-DOPA data showing that dopamine inhibits insulin secretion in a dose-dependent manner, and that this is conserved across species. Now, beyond that, I showed you a few slides ago that these beta cells, which secrete insulin, express all 5 dopamine receptors. So is it that this inhibition by dopamine of insulin secretion is driven specifically by D2-like receptors? So what we did was we took very specific agonists of D2 and D3 receptors, and in this case, quinperol. And what we found was that analogous to dopaminergic inhibition of glucose-stimulated insulin secretion, the quinperol replicates these data almost perfectly, suggesting that D2-like receptors do indeed play an important role in this inhibition. Moreover, beta cells are not the sole constituents of islets. Other cell types, including alpha cells, play important roles in metabolic regulation, in glycemic control, and they sit cheek by jowl next to beta cells. And these different cell types are in continuous communication. And in fact, transcriptomic analysis of human alpha cells and beta cells shows that, in fact, alpha cells themselves express D2 in addition to beta cells. So if you have alpha cells as well as beta cells expressing D2 like receptors, And we know, for example, that blockade of D2 receptors is a characteristic of antipsychotic medications, these psychiatric medications that cause diabetes. Is it possible that targeting these receptors can impair glycemic control? So what we did was we provided these antipsychotic medications to islets directly, human islets. And what we found was that if, as I just showed you in that last series of slides, dopamine and dopamine signaling physiologically inhibits insulin secretion, then one would postulate that blockade of these receptors would do the opposite, would raise insulin and glucagon secretion. And in fact, that's exactly what we see. In fact, we see clozapine raise glucagon secretion 300%, followed by olanzapine, which raises it by 200%, followed by haloperidol by 150. And we see similar increases in insulin. Now, the interesting thing here is that these increases, particularly in glucagon release from human islets, are in direct proportion to the metabolic liabilities of these medications. So clozapine classically is the worst offender in terms of induction of type 2 diabetes and dysglycemia, followed by olanzapine, followed by haloperidol. It seems almost too good to be true. Is it possible then that dopamine signaling is a common thread that runs through islets and islet communications and disrupting these communications by interfering with the ability of the local receptors to signal through this locally produced dopamine, therefore interferes with this very fine coordination between these different cell types in order to maintain glycemic control. And what I'm showing you suggests that at least some of these effects are in the periphery rather than in the brain, which these drugs were originally designed to do. Which goes back to the first question I raised at the beginning of the talk. What is it that is responsible for dopaminergic control or regulation of metabolism, central versus peripheral effects? This slide suggests that peripheral effects have some important contribution, but is it the main contribution? So that begs the next question. If antipsychotic drugs cause insulin resistance and diabetes by acting directly on islets to block islet cell D2 and D3 receptor signaling, D2-like receptors, can the converse also be true? Can agonism of pancreatic D2-like receptors treat dysglycemia? Can it begin reversing these effects? And just as importantly, how can we go about testing these important questions? So one way that we can start getting into this is with this FDA-approved drug that treats type 2 diabetes and its associated dysglycemia, bromocriptine. Bromocriptine is an ergot alkaloid compound that's been used for decades, at least the last 60 years, to treat prolactin-secreting tumors in the hypothalamus, prolactinomas. And it's a well-known agonist of these D2-like receptors. So we know qualitatively that people who have been treated traditionally with bromocriptine have an improved glycemic profile, which is what impressed the FDA enough to give it a designation for the treatment of type 2 diabetes. But the mechanism has not really been well described. Is it possible then that outside of its effects in the central nervous system, that because we know that these same targets are also expressed in peripheral targets like the endocrine pancreas, that the bromocriptine is in part having its beneficial metabolic effects through direct actions on beta cells? So we replicated more or less the same types of experiments we did with antipsychotic drugs, or dopamine for that matter. We took human islets and mouse islets, and we treated them with bromocriptine or with a known D2 agonist like quimperol. And in both cases, we had a very potent dose-dependent inhibition, and in fact, of glucose-stimulated insulin secretion. And in fact, the bromocriptine was several-fold more potent than this classic D2-like receptor agonist, quimperol. And when we took mouse and human islets, we saw, in fact, that looking beyond just glucose-stimulated insulin secretion, looking at glucagon, the bromocriptine was potent at picomolar concentrations in terms of its ability to lower glucagon. So by lowering a hormone like glucagon, which raises blood sugar, in fact, it begins acting at concentrations which are far below those that are necessary to elicit effects on insulin secretion, suggesting that the alpha cells may be targeted first by a D2-like agonist. And by lowering glucagon, you again start changing this intra-islet conversation, which gives us more clues, suggesting that D2-like agonists, such as bromocriptine, are acting through dopamine receptors on alpha cells and on beta cells. In alpha cells, they lower glucagon, which lowers hyperglycemia. In beta cells, they lower glucose-stimulated insulin secretion, which then causes the beta cells to enter a state known as beta cell rest. This is a concept which is increasingly emerging in the beta cell field. The idea that beta cells undergo stress in the setting of insulin resistance. By being pushed to their limits, this actually makes the cells less capable in the long run of being able to match demand. And by lowering insulin secretion, you not only resensitize peripheral targets, but also improve beta cell health, which in the long run reduces insulin resistance. So you have these twofold mechanisms, lowering hyperglycemia, which then diminishes overall insulin resistance, and so forth. So this then begs the next question. We can ask, can peripherally limited D2-like receptor agonists be used to block or even reverse antipsychotic drugs, which by definition block peripheral and central D2-like receptors? So if you want antipsychotic drugs to block their intended targets, which are in the brain, but not cause metabolic side effects through their actions peripherally on islets, for example, is it possible to couple these antipsychotic drugs with peripherally limited agonists so that they can compete out these targets which mess up insulin secretion and glucagon secretion? So this was the genesis of a series of studies where, working in collaboration with Dr. Amy Newman, the head of medicinal chemistry at the National Institutes on Drug Abuse, NIDA, and the head of intramural research at NIDA, Dr. Newman and her lab took classical D2-like receptor agonists and modified them chemically by adding a methionine group to make them more polar and therefore hopefully less permeable to the blood-brain barrier. So we took quimperol, we took bromocriptine, we took somanerol, we took well-established D2 specific agonists, and even partial agonists like aripiprazole. Then when we modified them and we examined their behavior, we saw for some of them a dramatic change in both potency and efficacy using functional assays such as a mitogenesis assay, which is used pharmacologically to test dopamine D2 receptor signaling. And what we began to see was that drugs like bromocriptine and their methionine derivatives were able to retain both potency and efficacy, suggesting that they might begin providing decent candidates to look at peripherally limited D2 signaling. So we took our top candidates, quimperol, bromocriptine, a drug called somanerol, and another drug called PD-128907, and their methionine derivatives. And we compared them now directly on their effects on glucose-stimulated insulin secretion. And what we found was, in terms of having the right mix of potency and efficacy, again, now directly on a functional assay like insulin secretion, bromocriptine seemed to deliver really well, as did its derivative. The only difference was approximately a 10-fold change in potency between bromocriptine and its derivative. And then we designated its derivative as our lead compound, and we called it bromocriptine methionide. So we then compared bromocriptine versus bromocriptine methionide in terms of its ability to directly signal at dopamine D2 receptors using an assay called nanobret, or bioluminescence resonance energy transfer. The main point here is this assay allows us to directly stimulate the receptors within intact cells and follow how well receptor stimulation is able to recruit either a G protein, such as inhibitory G alpha I1, or another intracellular effector known as beta arrestin 2, which controls the duration of receptor signaling at the cell surface. And what we found was that, very interestingly, bromocriptine methionide actually has preferential signaling. It recruits beta arrestin 2 with greater potency to the dopamine D2 receptor compared to even its parent compound, suggesting that, that this drug may have its own unique pharmacological properties. But nevertheless, it definitely confirms that it's capable of signaling through D2, similar to the unmodified version. So then, in collaboration with Dr. Barbara Slusher and her lab at Johns Hopkins, we started characterizing the pharmacokinetic and dynamic properties of bromocriptine versus bromocriptine methionide. And what we found in terms of pharmacokinetics was a pharmacokinetic profile which was comparable to bromocriptine. So within 30 minutes of exposure, both bromocriptine and bromocriptine methionide were largely metabolized. In terms of pharmacodynamics, what we saw was also quite interesting. Systemic administration of bromocriptine demonstrated basically very rapid breakdown. And what we found was that we saw within 15 minutes relatively robust amounts of bromocriptine in both the circulation as well as in the brain. In bromocriptine methionide, what we saw was less overall breakdown, and we saw continued or delayed permissiveness of this modified compound. So there was more that hung out in the periphery, which affected the brain plasma ratios. So it's not perfect. Now, granted, this is the first generation of these types of drugs, but there's something about this chemical modification, this methionide group, which delays access of the drug to the brain, allowing more of it to signal in the periphery. So at least it's a partial victory. So what is the relevance of this metabolically? Well, when we take dysglycemic black 6 mice which are fed on a Western-style high-fat diet, they develop insulin resistance and glucose intolerance, which is something that's very well established. And what we find is that when mice are given vehicle, you see classic profiles of dysglycemia in a glucose tolerance test. When you treat them with bromocriptine for several weeks, you see markedly improved glucose tolerance. This is the red curve compared to vehicle in these Western diet, high-fat diet-fed mice. When you give bromocriptine methionide, when you largely limit the exposure of this agonism to the periphery, you actually lose bromocriptine's beneficial metabolic effects. It resembles vehicle in this case. And you can see that according to the the blood glucose levels also under fasting conditions. So under fasting conditions, you also see a significant improvement in fasting blood glucose in the bromocriptine treated compared to both vehicle or the methionine. And the same trends follow with insulin tolerance testing, arguing that bromocriptine that's unmodified, a drug which has systemic access to both brain and periphery, is able to improve the metabolic profile in a way where beginning to introduce limitations to access cannot. Furthermore, we can then, instead of giving systemic access, limit bromocriptine to only the brain. So intracerebroventricular administration of a D2 agonist like bromocriptine eliminates its beneficial effect. So whereas I showed you a slide ago, the bromocriptine causes significant improvements in metabolic profile when it has access to both brain and periphery, shows that the converse is true. When you limit it only to the brain, you lose its beneficial effects metabolically, arguing that you need agonism, you need signaling at these receptors in both the brain and the periphery. You need tandem targeting in order to be able to satisfactorily achieve these effects. Now, in collaboration with Dr. Margaret Hahn's lab at the University of Toronto, we then asked whether the ability of providing dopamine precursors to the central nervous system is able to recapitulate what I just showed you with bromocriptine. And so intracerebroventricular administration of dopamine precursor L-DOPA similarly has no benefit, which reiterates the idea that you need both dopamine signaling in the CNS as well as in the periphery in order to change, to modify, to improve glycemic control. So the conclusions of the work which I've presented basically show that we've developed a new family of pharmacological tools with diminished blood-brain barrier capability, which can selectively manipulate peripheral dopamine D2-like receptor signaling. And the lead compound, bromocriptine methionine, shows preserved receptor binding and functional efficacy, suggesting that this can be a useful tool. And with that in mind, we can use a tool like bromocriptine methionine in parallel with its unmodified parent molecule, bromocriptine, to begin dissecting the relative contributions of dopamine signaling in the central nervous system versus in the periphery in terms of better understanding the mechanisms as well as the treatment of dysglycemia, such that when you systemically administer a dopaminergic D2 receptor agonist like bromocriptine, it can improve dysglycemia because we know it's hitting targets throughout the body, brain and periphery, but that these metabolic improvements can be attenuated by restricting access to one or the other compartment through these chemical modifications, or by limiting the access of the drug to its targets through targeted administration, which leads us ultimately to say that the tandem coordinated actions of both brain and peripheral dopamine D2 receptors are required for correcting dysglycemia. And with that, I'd really like to thank so many people I've had the privilege of working with, including members of my lab, like Despina Aslanoglu, Zach Farino, Fong Li, Caroline Ward, Shinaz Bano, Amy Newman's group, Margaret Hahn's group, Gary Schwartz, Rita Bettino, and my funders. So thank you.
Thank you so much, Zach. That was fascinating and really interesting how you methodically went about sort of carving out the true role of the different pieces and how they connected to the D2 receptor. I had a couple questions for you. I'm going to invite people to unmute themselves or drop questions in the chat. But I also had a question for you. With these D2 receptors, do you guys ever look at how they are impacting or waxing and waning impact in immune cells? in concert to this whole metabolic landscape?
I think that's a wonderful question. So, the more that I look at this receptor system, the more I realize just how ubiquitous it is. So virtually
Mhm.
every major system in the body is capable of taking up these amino acid derivatives and converting them into dopamine, or in the case of tryptophan, turning it into serotonin and so on. In the case of dopamine, you're absolutely right. Immune cells can make their own dopamine, presumably, because they express the biosynthetic machinery, and they express these dopamine receptors. What the receptors are actually doing immunologically is really not understood at all. Are
Mhm.
they acting in an inhibitory manner is really unclear. But I do believe that it does have important implications. And it's possible that, at least under physiological conditions, local islet dopamine is somehow communicating with the local immune cell population to regulate cytokines, and vice
Right. I
versa.
mean, that's why I brought it up, because we're all about obviously the destruction of the islet beta and alpha cells, sometimes mostly the beta cells in the islet during the progression to type 1 diabetes. But
Yeah.
we know like dopamine D2 receptors are found on several kinds of immune cells, CD4 T cells, microglio, macrophages, all of those who participate in or are thought to participate in the attack of the beta cells in type 1
Mm.
diabetes progression. But if you think about it, like I know from some of my neuroscience background, like I believe clozapine, isn't it true that the patients also undergo like some kind of decrease in white cell count?
That's
And
exactly right.
right, and
Yes.
so is this being mediated by these D2 receptors? Are those getting knocked out? I mean, it's really fascinating.
No one knows. It's idiopathic. And that actually is a big thing for people taking clozapine, because they need weekly blood draws, because what happens is that they call it idiosyncratic neutropenia. So
Hmm.
their white count is decimated all of a sudden. You can have people on these medications for years and they're stable, and then out of the blue their white count starts dropping for reasons that we really don't yet understand. Is that
And at
related?
the same time, are they, when that happens, do they enter the dysglycemia? Also, do they get type 2?
So that's a great question. The you know typically, uh, you know people rapidly develop dysglycemia on clozapine, and that, I believe, is independent of whatever is happening in terms of white blood cell count.
Hmm. Yeah, but they both have these D2 receptors
Yes.
um, involved in those different cell types. Did you have a question, Maggie? Oh, no.
Yeah, I do. Nice, great talk as usual, Zach.
Oh, thank you.
I always learn something from your talks, even though I've heard you
Too
many.
many times. I'm sorry.
I was just wondering, I'm fascinated by the fact that the, glucoregulatory effect is lost when you add the MEL um, additive or
Mhm.
so what do you think the brain contributory mechanism is? Because L-DOPA in the brain doesn't seem to do anything, bromocriptine in the brain doesn't do anything, right? But if you incubate the cells with these drugs, then something happens. But like, what's your hypothesis? That it just intrigues.
Sure. No, I mean, so the majority, we know for a fact that interfering with dopamine signaling in the hypothalamus, and in particular in the arcuate nucleus, in POMC positive cells, has a profound effect on metabolism. And you can induce dysglycemia by interfering with the function of these cells. It's possible, because they intersect with the circadian framework, that there is something about nutrient sensing that is important in more global function. These cells project to the peripheral organs. They project to the pancreas, to adipose tissue, to the gut, and so on. This might be, so rather than having a direct effect, what they might be doing is they might be helping to coordinate. And I think that by untethering them when you start having all of these systems operating more autonomously, that absence of coordination then leads to miscommunication that over time causes a drift in blood sugar control.
Yeah, and then is there hope for psychiatry to develop something? Because if we still need to block D2 in the brain, then, like, are we stuck? And is bromocriptine safe maybe in schizophrenia? Has anyone looked at that? If someone is on an antipsychotic, can that override the safety effects? Like, and because I know like Gary Remington, he's
Mhm.
done L-DOPA studies where people who are stable in antipsychotics, he gives them L-DOPA.
Mhm.
And these are like really small, I think he looked at like 12 people.
Mhm.
Um, and it seemed to be in this, again, uncontrolled setting, seemed to be safe. But is that something we should be testing, or has that been tested? Like, is bromocriptine safe?
So yeah, that's a wonderful, and that's actually a clinical trial that we're trying to get off the ground. Um, but your question, as usual, is brilliant, and essentially the answer is yes. So there was a period of time in the 1980s when people were thinking about how to more effectively treat tardive dyskinesia, which is a cluster of motoric side effects of long-term dopamine receptor blockade in the striatum in the brain. And people thought that giving agonists of these receptors would help reset their function. And these were clinical trials which were run by John Kane, who was one of the people behind the development of clozapine, and Jeffrey Lieberman. And what they found actually was that people chronically treated with antipsychotic medications did not become more psychotic. Their psychiatric symptoms were not exacerbated by co-administration of bromocriptine alongside long-term antipsychotic drug use. But not only that, their cognitive symptoms actually seem to improve. And that's very exciting.
Yeah,
Actually.
and that was my next question. Cognition and negative symptoms, right?
Yes. Yes. It actually helped tune it better. It toned them up. So, and what I'm proposing then is, while we're, working, knock wood, on the development of hopefully, subsequent classes of these peripherally limited agonists, I think a small subtherapeutic dose of an agonist like bromocriptine could be co-administered safely, because the majority of whatever risk is going to be absorbed by long-term blockade of the receptors in the CNS. Whereas in the short term, especially if you take the drug orally, the majority of say a 5 milligram dose versus a 20 milligram dose is going to hit peripheral targets.
I think we have to do this trial, Zach.
Yes.
Well, hats off to both of you in terms of driving this forward. I think it's an understudied region. Not sure why, but it does seem to be yielding other fruits like things that could, that are metabolically important in terms of the knowledge base for those who study type 1 and type 2.
Mm.
And maybe this patient cohort might be interesting for people to sort of dive into. Again, those that study type 1 and type 2 diabetes, looking closely at how their metabolic framework, changes in context of these drugs or without them. And I just think it's a really interesting place that we don't often hear from. So thank you so much for unpacking your work, and really great to have you today. We can't wait to see what comes next from the Freyberg lab.
Oh, I'm so grateful, and thank you, and thanks to Dr. Han, and I see that Dr. Zhang is here as well, who's a wonderful collaborator, and to the greater community. I think there's a lot of exciting stuff, coming, and it's really an honor and a privilege. So.
That's great. Well, have a great rest of your day. This video recording will be housed on our ever-growing library for those who missed it, and they can watch it at a later date. Have a great rest of your day, everyone.
Take
Bye-bye.
care. Thanks again.
Bye-bye.
Yeah, bye.
Thank you.